So I already have a pretty good explanation for you of mitosis, and a lot of places people will easily find information on mitosis. But then you're often asked to compare mitosis and meiosis. It's one of the things that typically as a four-year biology student is finishing their degree and they take an exam and they're asked certain things about photosynthesis or animal and plant cells and DNA and RNA and mitosis, meiosis. Most people don't, they get messed up on specific things. So I want to try to go over just the key elements of meiosis. I'm going to try to relate it specifically to how they're going to be used, how understanding the process is useful as we do genetics problems, the Mendelian genetics problems. In addition to that, I'm going to create a second lecture information on really what the purpose is, which is gametogenesis. So meiosis is a type of cell division. It's a unique type of cell division. It's a type of cell division that only occurs by germ cells. So all the cells in your body are not capable of carrying out meiosis. They can all carry out mitosis. That's why it's important to learn about mitosis. All cells, plant cells, animal cells, all the cells of the body, all the cells of different organs and tissues can carry out mitosis. And it's an important process for growth and development. However, meiosis is unique. It's a process that's only used for gametogenesis. So it's the process that creates the gametes, the sperm and the egg. So there are two sort of sub-processes that go into more specific details. That's called spermatogenesis, which is how this process results in the creation of the sperm, and then oogenesis, how this process ends up in an ova, right, or an unfertilized egg. So that's something I'm going to go over, but not right now. We're going to go over the basics of terminology and the overall process and unique parts of meiosis. So, terms. Chromosome, right? So chromosome is going to be our DNA and its associated protein. Now when you see a chromosome, you're usually seeing some image that looks like this, because that's the only way you can usually see physically a chromosome under a microscope and that photographs are taken of it. Unfortunately, this isn't just a chromosome. Technically, this is two chromosomes. So what you're really looking at here are two chromosomes that should be identical to each other, and they're called sister chromatids. So the chromosome is our DNA, and then that DNA is organized with proteins, right, into the chromosomal structure. Right? So you have underlying DNA structure, double-stranded DNA, and then DNA replication occurs, right? So when DNA replication occurs, you get two, right? You typically, right, we have this kind of idea, right? We have the A, T, G, C, C, T, A, C, G, G, are two strands of DNA, right? They serve as templates. T, A, G, C. I'll make it really simple here. T, A, C, G. Like that, right? And then there's two new DNAs that are created. And there's all kinds of details. You know, we go over this in lots of detail in class. Oops, sorry. Upside down. T. Yeah, it should be upside down. All right. So you get the idea. Two new double-stranded DNAs are created, except most of your studies of this kind of lets you think that they're just separate, that they just now the process is done and they're two new but they're not. They're connected together, right? So there's a cohesin protein that actually holds these two new double-stranded DNAs together, right? And so they are in chromosomes, which are held together as sister chromatids. They will only divide during the cell division process, during the mitosis process or the meiosis process. So for either process, mitosis and meiosis, what is similar? Starting off, DNA replication precedes each of them. So the DNA, the chromosomes in the cell will double and you'll result in sister chromatids for both mitosis and meiosis. So a germ cell is 2N. Okay, that means here, not have two different colors, these are two different chromosomes. Here, these are two versions of the same chromosome. That's what we mean 2N, right? If it's 1N, then you can't, I'm blocking enough to me, not to you, but you would just have two different chromosomes and not two copies. Right? So in this case, you have these chromosomes here, which are called homologous. Now the idea here is that those homologous chromosomes, one of them came from your mother, let's say, it's maternal, and one came from your father, it's paternal. And so while they have the same genes, like a gene for the enzyme in glycolysis hexokinase, maybe that gene is on this chromosome and on this chromosome. So you have a version from your mother and a version from your father. Typically only one of those will be expressed, right? But you'll have them on both chromosomes. And so forth. All your genes, you essentially have these two copies of them, one from your mother, one from your father. So each chromosome has them, and then you have two of each chromosome. So that's why those cells are called 2N cells. All right, hopefully that makes sense. So we have two separate things here. People get these confused constantly. Difference between sister chromatids and homologous chromosomes. Homologous chromosomes are chromosomes that have the same genes but potentially different versions of those genes. Now, when we later do the problems, like genetics problems, we might say brown hair is dominant to, let's say, black hair. This is just an example. Doesn't matter, the organism or whatever. And so what we would do is we would write like a big B like this, and that would be brown. And then a little b like this, and that would be black. So they'd represent them. And so what that means is that, let's say, the enzymes that code for the pigment that would then result in this particular color, maybe on this chromosome you would have the big B, and this one here you would have the little b. Okay? So the idea is, while these chromosomes carry the same gene, say hair color, they can have different alleles for those genes, different versions of the gene. So those are the homologous chromosomes. The sister chromatids, they should be the same. So if this one had the allele for brown, then this one should have the allele for brown, because they should be exact copies. They are the result of DNA replication. Okay, so sister chromatids should be, aside from mutation, exactly the same. All right, hopefully that makes a little bit of sense. Okay. So now we start to get into the process a bit. Now, if you've studied mitosis, which hopefully you have, we have different phases, all right, in mitosis. All right, so we have our interphase, which is where, when you look at a slide of mitosis, you pretty much see most of the cells in interphase. That's the phase where all these other cellular processes are taking place, getting ready for division. So things like DNA replication and protein synthesis, gene expression, that's when that's happening, getting ready for the process to occur. Once we enter prophase, what happens is that these chromosomes are big. So if these are supposed to be cells I'm representing here. So let's just say this is one of these cells. The chromosome, the length, the size, I drew these little tiny ones here. The length of a chromosome, if it is not condensed, can be as much as 10 times the length of the chromosome. So this is chromosome length. Can be 10 times the diameter of the cell that it's being housed in. And now remember, these chromosomes are not only being housed just in the cell, they're being housed in the nucleus of the cell, which is then going to be a much smaller compartment. So the thing is, the chromosomes have to be condensed, right? And if they're just these long stringy structures and they're not organized in any way, then they're just going to get all tangled. So proteins, all right, will help them. Okay, so there are histone proteins. That's another sort of separate topic. And then here we have condensins. Condensin is going to make them visible to us, but that's not the point. They don't care whether we can see them or not. It's so that they are organized in the tiniest package possible for division. So they can divide into new cells without being tangled or without being confused or getting bits of them here and there, condensing them into nice little packages. So that's what's going on. So that's happening in prophase, condensing. And so they start to become visible. Now in these cells too, DNA replication is over and they're starting to condense. Again, you're going to have now the sister chromatids. Two, of each, like this. Okay. Then you get into, well, I usually talk about in my classes the pro-metaphase. Most people skip it and they just go right to metaphase. But really, it's an important step. It's a step where the nuclear envelope starts breaking down, and it's where initially the microtubules, the kinetochore tubules, those are involved in actually separating the chromosomes or sister chromatids from one another, actually start to attach here, right, to the centromere, to this particular part of that sister chromatid pair. That's what's going on in prometaphase. It's very hard to see. Usually people don't see it very well under the microscope slide, so people tend to just skip it. But it does happen. It's an important part of this whole procedure. We then get into metaphase, and this is what I'm going to talk about now, because this is something that's really important for what moves forward and how meiosis is different than mitosis. All right, so metaphase. This part all kind of similar, mitosis, meiosis. Now metaphase in meiosis, I refer to this now as metaphase 1. Normally what happens in mitosis is that all these chromosomes would be lined up then in the middle of the cell. Now they don't line up themselves. They can't do anything. They're just sitting there. They're inanimate. All right. But there are microtubules coming out of a microtubule organizing center, and they're attaching. And then what they do is they start to pull. So they start to pull, and they're trying to pull this set of sister chromatids in this direction. And on the other side of the cell are microtubules trying to pull them in the opposite direction. But they can't be pulled apart because the cohesin's holding them together. So instead, that tug-of-war ends in a tie, and we get what we call metaphase, like this. Now, what's different here is in metaphase of meiosis, we end up getting the homologous chromosomes pairing up. Like this. So you can see metaphase of mitosis metaphase, metaphase of meiosis, which we would call metaphase 1. So in metaphase 1, there's this particular characteristic. It is the pairing of homologous chromosomes. Now, the thing is, I said here, one of these could be from the mother, one could be from the father. This is where we start to get variation. One of the points of meiosis is to create almost infinite variation in these gametes that are eventually going to be produced. So say there's 4 sperm that's going to be produced from this one germ cell. They're all going to be unique from one another. Every single germ cell that will produce these 4 sperm, they're going to be different. And they're going to be different, they're going to be different, they're going to be different. There's going to be an infinite number of combinations. That's partly going to be, starting off right here, is because you could say, okay, so that means this side over here, this side could be the 2 maternal ones, and this side could be the 2 paternal. So we could separate them and have one cell, eventually with the 2 maternal chromosomes, for example, like this, and one cell with the 2 paternal chromosomes, like that. So these would be the two ones from the father, 2 from the mother, for example. Or they could have switched here. In this particular process, they don't have to line up a certain way. So you could get, this one could be the mother's, and this one could be the father's. Vice versa, father, mother, or switch the other way around. So you get, that's one sort of variation, but that's not infinite. There's only a few possibilities. Where it becomes sort of an infinite sort of bunch of possibilities is what's really happening here. So again, in metaphase 1, this is an important phase, okay, to talk about. is what we're also going to see is the process called synapsis. Crossing over, also referred to as genetic recombination. So there's a lot of terms for this. So they're all really referring to the same thing. What we mean here is this. When these homologous chromosomes pair up, okay, I'm exaggerating because of the size here. Some of the sister chromatids, one from one pair, can actually overlap with one from the other. And let's just put a different gene on here. Let's have I don't know, something that's distinct. Let's say I'll just put a Q, capital Q here, and that means both of these would have capital Q. And on this one I'll put little q. Okay, so that's just a gene, right? So the father gave the little q gene, the mother gave the capital Q gene for whatever that happens to be coded for. She gave the gene for the dominant, he gave the gene for the recessive. Now what's going to happen here in the crossing over is that her gene for the dominant is going to cross over with his gene for the recessive. And before this separation takes place, what's going to happen is they're going to swap. They're going to actually swap chromosomes. And so what that means, if I just pull out these, that means here, here, here. That means this chromosome here from the mother, which has all maternal genes, had two. One had the dominant Q, the other had the dominant Q. But now crossing over has occurred. There's been a recombination, a shuffling of the genes. And so now genes from the father's chromosome have joined the mother's. So you have mother's genes here and father's genes on the exact same chromosome. Here, the mother's gene has joined the father's chromosome. So now you have one chromosome that's completely maternal, one that's a hybrid. So if I say I'll color code these a little bit different, let's just say, so this one is all maternal. This one is all maternal with a little bit of the genes from the father. I'll recode this here. This one is all the genes from the father. And this one is mostly genes from the father, but now it's received some genes from the mother. So you can see we actually have 4 different versions of this chromosome, as opposed to just the 2 versions. Now the thing is, that's one crossing over event. Crossing over can happen multiple times on a chromosome and in all sorts of different locations along the chromosomes, which essentially means any of the genes pretty much between the 2 homologous chromosomes can be swapped to make these hybrid chromosomes like this. So this is a very important part of meiosis. This does not happen in mitosis. Mitosis doesn't see crossing over because these never pair up. Right at the beginning in metaphase, they're separate from one another. Okay? In meiosis, they pair up like this, so you then get the crossing over occurring, and then you get the shuffling of genes, and you get new, different variations. So that will happen, and that's kind of one of the most important things really about the whole process of meiosis. The other thing that's important is then we're going to get division. So you can see they divided here. So metaphase occurred, they paired up, the homologous chromosomes paired up, there was shuffling of genes, recombination, and then eventually they separated. So a signal is going to be sent to release that cohesin so the sister chromatids can be pulled apart as separate chromosomes, except in this case that doesn't happen. Right? We don't actually pull them apart. What we're pulling apart are the homologous chromosomes.